Hi, my name is Elliot Hagedorn. I'm an assistant professor in the HEMONC section here on the Med campus. My lab is broadly interested in understanding how cells move between different tissues, which is something that's important for normal development and physiology, and is inappropriately activated during numerous disease states. There's a lot of therapeutic interest in understanding how cells move across tissue boundaries, inhibiting this process in contexts like cancer metastasis. And there's also interest in promoting or stimulating cell migration, for example, for the various cell-based therapies that are currently being used to treat cancer. On the left here, I'm showing a schematic of the human vascular system. What I find fascinating about this system is just how extensive the blood vessel network is. It reaches to within 50 to 100 microns of every cell in our body, such that any cell in circulation can theoretically reach any tissue in our body. What's not shown here is that the endothelial cells that line these blood vessels can vary dramatically depending on the tissue or organ. My lab is interested in understanding this vascular heterogeneity with the idea that we can exploit this or manipulate this to get circulating cells into target tissues. Our approach involves using the transparent zebrafish embryo, where we can directly visualize circulating cells and the vascular endothelium in different tissues. One of the tissues that we study is the hematopoietic stem cell niche, which is in the tail of the embryo. I'm showing here a still from a time-lapse movie where we can see two green blood stem cells coming in through circulation. These cells will specifically adhere and interact just with this vasculature in the niche tissue and then crawl across the endothelium to access their niche in the extravascular space. In our recent work, we generated a transgenic zebrafish line that allowed us to isolate just these niche endothelial cells and compare them to the endothelial cells of other tissues. In doing so, we identified a cohort of genes that were selectively expressed by these niche endothelial cells. This included molecules with functions regulating cell-cell adhesion, as well as endocytosis and membrane trafficking. The cell-cell adhesion bit made sense given the adhesive interactions we had seen in our time-lapse imaging. The endocytosis expression signature was unexpected. To explore this further, we injected an endocytosis reporter into circulation and confirmed that indeed the endothelial cells in this hematopoietic niche tissue are extremely endocytic. At higher resolution, we can see that each endothelial cell in this niche tissue contains a discrete cluster of endocytic activity, and it's at these sites that the blood stem cells will specifically adhere to and transmigrate the endothelium. And so what we're thinking about based on our data is that these endocytic clusters serve almost as portals for transmigrating cells across the endothelium. We're now using the tools in the zebrafish to knock out candidate genes that we think are regulating this biology. We're also using tools to ectopically express this machinery in other endothelial cells with the goal of getting cells to stick and transmigrate into those tissues. To translate our work in the zebrafish into a human system, we've been collaborating with Chris Chen's group on the Charles River campus using their biomimetic human blood vessel models, where we can line these microfluidic chips with different types of endothelial cells from different tissues and flow through different blood, immune, and cancer cells. And then just at the end, I wanted to put a plug in for the zebrafish as a really great system, an in vivo system for collaborating. It's very easy to transfuse different human cells into the zebrafish. And there are also a number of great primary tumor models in the zebrafish. And I'd be happy to talk further about collaborating if anyone is interested. Thank you.